Okay, now what we learned just now, or last 2-3 hours, when talk about gene transfer, we didn't do editing. Are you clear? Gene transfer, basically we take the gene from first organism, transfer it to the second organism. So this is meant by gene transfer. Okay, meant by gene transfer. So do we edit anything? We don't edit the gene. Okay, but in gene editing mechanism here, what we can do is a type of genetic engineering, which DNA we can insert in DNA, we can delete the DNA, we can modify the DNA, and even replace the DNA. Okay, so in two conditions, we need to do this. First, if a person, okay, have a disease caused by a particular gene, so what we can do actually, we can delete this gene. Okay, we can do gene knockout. So as long as no that particular gene or particular allele, so no disease. Are you clear? Okay. Or sometimes the disease is caused by dominant allele. Okay, dominant allele. So what we're going to do here is we can edit this dominant allele. Can I say that? Either we inactivate a particular gene or we can edit the dominant allele so that it won't cause a disease caused by dominant caused by dominant allele. What we can do, we can edit it so that it becomes a recessive allele, then the disease won't appear. So we can do the gene editing. Gene transfer cannot help us to do this. Gene transfer cannot help us. So what we're going to do here is this could use for targeting mutation. We purposely mutate the gene so that the gene become inactive. Or in gene therapy, we can insert the functional allele. Let's say, for example, the allele can't function, we insert a functional allele so that we can cure a particular genetic diseases. Okay, but before the discovery of the CRISPR-Cas9 system, the whatever method, the genome editing in the eukaryotic cells, okay, is possible by using various methods, but it's very inefficient and impractical to implement it for large scale until the discovery of the CRISPR-Cas9 system. Okay, which is highly selective. I will tell you guys, in terms of the mechanism later. So CRISPR-Cas9 is a very, very new technique discovered in 2012. Okay, it's a very, very new technique where we can program this system to help us to edit the genomics DNA. Okay, the genomic DNA. So what's mean CRISPR? CRISPR stands for clustered regularly interspaced short palindromic repeats. Okay, and Cas9 stands for CRISPR associated protein 9. Okay, so how this system actually do? In general, okay, bacteria capture and snips the DNA from the invading viruses and use them to create the DNA segments known as CRISPR array. So it's something like an immune system for the bacteria. So this CRISPR array allows the bacteria to remember. It's something like our memory B cells and memory T cells. It can remember that particular virus or closely related one. So if the virus infect the bacteria again, the bacteria produce these RNA segments from the CRISPR array to target the virus DNA so that before the virus give any effect to the bacteria, so they have this enzyme called Cas9 to cleave the DNA apart, disable the virus. Okay, so now the entire system is something like this. Now look at this. So this is the bacteria. Okay, the bacteria. So at the DNA there, you can see that bacteria have this what we call the CRISPR array. So what's mean CRISPR array? CRISPR array consists of the repeat. Okay, so this one is a repeat. The DNA repeat. Let's say APCG, 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 repeat. In between the repeats, they have this what we call the spacer. They have this what we call the spacer. And when the scientists analyze the spacer DNA, their DNA, are you clear? Their DNA, maybe this is ATCG, for example, ATCG, repeat, ATCG, ATCG, keep on repeat. In between this ATCG, they have this what we call the spacer. So the spacer, let's say for example, in this case, AAA. This one CCC, for example. Okay, for example. So the spacer were different. Spacer totally different. Are you clear? And when the scientists analyze the sequence of the spacer, they realize that this sequence actually from virus DNA is similar or exactly the same as the viral DNA. So scientists wonder why. Why in the sequence of the bacterial appear viral DNA? And nowadays, scientists realize that whenever there is bacteria infected by the virus, so what virus normally do? First thing, they're going to release their DNA. Okay, so what this bacteria are going to do is they take this DNA, part and the pieces of this DNA, and make it or incorporate it into this spatial region. Let's say, for example, this one TTT. Can you see that? And then they have to repeat again. So it's something like records, the records of what kind of virus have been infected the bacteria. Are you clear? So these are the spacer. So what's so important about this spacer? Okay, what's so important about spacer? Now, if the same virus infect them again, same virus. So what will happen here is virus will release their DNA. Okay, this viral DNA. So what happened here is this spacer will be used to code for the single guided RNA. Are you clear? Use this as a template to code for single stranded guided RNA. So what's this function of single guided RNA? Okay, now you look at this. What will happen here is we do have the enzyme. This enzyme called Cas9. So this is what Cas9 actually do. Cas9 will take this single-stranded, single-stranded RNA. We're going to take here. So the strip. So this is a single-stranded guided RNA. So what's the function here? So they try to match. Can I see that? So what's the Cas9? Cas9 basically unwind the viral DNA, match it with this guided RNA, so that when they match, they're going to cut the viral DNA. So it means that they're going to make the viral DNA become fragmented. When it fragmented already means that viral DNA is actually break down the viral DNA, so viral DNA will no longer cause the problem to the bacterial cells. Do you get the idea? So basically, it's a protective mechanism. It's something like immune system, the memory, okay, the immune system. So first, in this bacterial DNA, scientists realized that between the repeats, there is a part called spacer. So this spacer, okay, when the scientists check the sequence of the spacer, scientists realize that this sequence belongs to the viral DNA rather than the bacterial DNA. Are you clear? The sequence? Okay, so scientists wonder why between this bacterial DNA that are the sequence of the viral DNA, between the bacterial DNA, why we have the viral DNA, and scientists now realize that it's because of the infections. So when the virus infect the bacteria try to take this viral DNA and incorporate into the spatial regions here. So when this virus infect the bacteria second time, so this spacer will be used to code for the single guide RNA. So this single guide RNA will bind to the Cas9 protein so that they can do the checking. Can do the checking. So when they do the checking like this, then what we're going to do, they're going to cleave this viral DNA. Okay, so an analogy with like something like the suspect. If let's say now you want to arrest a suspect, you don't have photo, you don't know. Can I see that? You don't know. So this single guide RNA is something like a photo of the suspect. Photo of suspect. Cas9 is something like a police. So how to arrest? If the Cas9 want to arrest or to kill the suspect, okay, or to kill, that means to arrest the suspect. So first thing, they need to have the photo of the suspect. So single guide RNA will act as the photo. Can you see that? So let the Cas9 going to do the job to search for, oh yeah, this is the viral DNA. So what they're going to do? They're going to cleave at the viral DNA. Okay, they're going to cleave at the viral DNA so that they're going to break this viral DNA. So it means that viral DNA no longer intact. Therefore, viral DNA won't be able to instruct. Okay, won't be able to instruct the bacteria to produce the proteins for the virus. Can you get the idea, the overall idea? This is the natural mechanism. It's not a man-made mechanism, but it's a mechanism naturally happen in bacteria. So now, why we are going to learn this? So what is this? What is the future of this technology? Try to imagine now. If let's say this is the human DNA, and we do know that these parts of the human DNA, the gene, cause disease. It causes disease. So we want to inactivate this gene. So what we're going to do here is, we do know the sequence of the gene. When we do know the sequence of the gene, what we're going to do here is, we take this sequence of the gene and we're going to synthesize guide RNA or single guide RNA. So when we synthesize a single guide RNA, and then we put this single guide RNA into the Cas9, then Cas9 will come to human DNA here and cleave the gene. Can you see that? They check. Okay, they're complementary, they check, and then they cleave at the human's DNA. Are you clear? So when they cleave the human DNA will have, because in the human DNA will have DNA repair mechanism. So DNA repair, it can be two-way. Okay, the first way is the we call non-homology, okay, end joining. Okay, non-homology end joining. Oh, sorry. Later I will show you. So what does this mean? Because DNA repair mechanism, you try to imagine when you break the DNA already, double-stranded. So DNA repair mechanism don't have any template. So in order to fill up this gap, so what will happen here is your DNA repair mechanism simply add the nucleotide. So when it simply add a nucleotide, it means that now they inactivate the gene. Are you clear? So in this process, or what we can do here is we can give them a direct targets. Okay, for example, now it break into two, double break, because cut by the Cas9 already. So we can give them a template. We give them a template so that they can form. So we can do the corrections here. So it means that we change the disease-causing allele to the non-disease-causing allele. Are you clear? So by using these two methods, we will be able to do the edit. So either we inactivate the gene or we do the corrections onto the gene. So this is what Cas9 can do. Okay, so come back to our notes here in detail now. So we just now we talk about CRISPR array. CRISPR array actually consists of the repeats and also the spacer. So repeated sequence of nucleotide are distributed throughout the CRISPR regions and interspersed with this viral DNA, the spacer. So you look at this diagram, you can see that repeat, repeat, repeat. So then you have the spacer, spacer, spacer. The spacer resemble the viral DNA. Okay, so what's Cas9? Cas9 actually is endonuclease enzyme. So write down, endonuclease means that they have the ability to cleave DNA at specific regions. It's something like our restriction enzyme, but this is a different form of the restriction enzyme. Okay, because it's a restriction enzyme, then it's very, very specific. Cas9, it will follow the single guide RNA. Okay, So you can see a Cas9. What's the function of Cas9? Cas9 first thing unwind the foreign DNA. Can you see that this is foreign DNA? So you unwind the foreign DNA. Can you check the gray color? Gray color is the foreign DNA. Okay. So then they have this, what we call single guide RNA. Can you see that? The blue color. So single guide RNA will have the sequence that complementary to the foreign DNA. So one it match already, can you see that? GCAT, GCAT, whatever. So can you see that? When they match already, what will happen? They're going to cleave. Can you see that? Where it cut? It cleave at the foreign DNA, break it into two parts. Are you clear? So this is enzyme. So this enzyme, if with itself alone, when alone they can't do anything, they must have this single guide RNA. So this single guide RNA will be the one that directs the Cas9 to cleave at the correct part, so that it's going to break the foreign DNA. Okay. So one single guide RNA? Single guide RNA actually got two parts. So single guide RNA actually got something like this. Got two parts. So this part is known as the trans-activating RNA. It's the part that binds to the Cas9. And CRISPR RNA is the one that we're going to match with the foreign DNA. Okay. So trans RNA are base pair having the stem loop structure and itself attached to the endonuclease, the Cas9. So this part trace, can you see that? Trans-activating CRISPR RNA. So this one actually is the one that binds to the endonuclease enzyme Cas9, and we have the CRISPR RNA is the one that later match with or complementary with the foreign DNA, so that later the enzyme can cut. Okay, the enzyme can cut here. So can you see that? This is the how this gene editing can be done by CRISPR-Cas9. You can see this red color is the guide RNA. This blue color is the Cas9. So Cas9, what you do? So first Cas9 unwind. Can you see that? Unwind the DNA. Okay, unwinds the DNA. It can be human's DNA, it can be the viral DNA, doesn't matter as long as they are DNA. So unwind. So once unwind already, then they use this guide RNA or the CRISPR RNA here to match the sequence. One is matched, then you're going to cut it. So it produced what we call the double-stranded breaks here. Okay, double-stranded breaks. So we can have this what we call non-homologous or non-homology end joining. Can you see that? The break, our DNA repair mechanism try to repair it. But once it repair, it can delete some of the nucleotide, it can add some of the nucleotides. So this one, it creates the inactivations of the, so this one going to inactivate. Inactivate what? Inactivates the gene. Or we can have this homology directed repair, HDR. So this homology directed repair, what happened here is we can use a donor DNA as a template, making and can do repair. Or initially it's ATCG here, then we change it to CCAT, for example, to correct. So basically this one we do the gene editing. Okay. So what is the first process? First process, so researchers create the single guide RNA with a short sequence that complementary to a specific target sequence of DNA in the genome that we want to edit. And then this single guide RNA bind to the Cas9 enzyme. So we use these single guide RNA to recognize the target DNA sequence. So it will be very, very specific. Okay, it will be very, very specific. And then the Cas9 enzyme now cleave the DNA at the target sequence, create the double strand breaks, and DNA repair mechanism now either add or delete pieces of genetic material by non-homologous end joining, or to change the DNA or edit the DNA by replacing existing segment with the customized DNA sequence through the homology directed repair. Okay, so with this, I have done for the gene editing. I'm going to show you guys the video clips, okay, regarding to this Cas9, okay, CRISPR-Cas9, how the process takes place. Okay.